All right, I'm going to be doing the Epstein-Barr virus for my presentation. So the Epstein-Barr virus, it infects humans, and the tissue tropism is B cells or white blood cells and epithelial cells. However, it's most common in B cells. So the portal of entry is orally. As a lot of you know, Epstein-Barr virus is mononucleosis. So you get that from sharing water bottles and whatnot through saliva. So the host cell receptor for EBV is this CD21 receptor. Let me get on the pointer. So this CD21 receptor binds with the glycoproteins GP350 and GP220. And what happens is there's a CR2 receptor, which you'll see in the next slide, which basically pulls the glycoproteins in towards the host cell before the spikes of the EBV virus will attach to the host cell. So here we have another picture of the viral entry into the host as it enters into the cytoplasm. Here you can see the spikes of the Epstein-Barr virus and the CD21 receptor here. So what's going to happen when it enters, it's going to form this tegumented capsid, which will be the tegument here, the viral envelope, and then here we have the genome. So it enters into the nucleus, which we'll get to later, and it's usually linear DNA, and then it's going to circularize, and through some processes, it's going to undergo chromatinization. Some proteins are going to acetylize the chromatin and make it better for transcription. So here we have viral entry again. Here you can see all the glycoproteins and the virus. We've got GP350, GP220, GHGL, GP42, and GP. Here we can see better the CR2 receptor, and it kind of moves in as it connects to the GP350 protein, and eventually it just moves it all the way to the membrane before the virus enters the cell into the cytoplasm. So the classification of EBV, it's a herpes viridae virus, which is Baltimore classification 1, double-stranded DNA. So the host cell and lytic infections will produce the mRNA through DNA polymerase. However, most replication is going to be in latent infections, which primarily uses the virus, the proteins. So the virus will produce some immediate early proteins, BZLF1 and BRLF1. They're both early proteins. BRLF1 is the promoter. I think an important thing to say about these two is that just one of them doesn't do much, but in combination, they're very good at replicating the genome. But they also have some different functions. BZLF1 can also inhibit apoptosis, and it is also responsible for acetylizing the chromatin. So here we have the viral replication, and here it's in the linear form on the left, and then here's a chart of the overall process. So what's going to happen is the genome has these tandem repeats at the end, and what they're going to do is they're going to associate with one another, and this is going to cause a circularization into an episome. And then we're going to have the rolling circle mechanism. So here this says ZTA. I know it's hard to see, but ZTA and BZLF1, from my understanding, are the same thing. And what's going to happen is the BZLF1 is going to cut the DNA into single strands as it produces the DNA. So here we've got latent replication. As I mentioned before, the immediate early proteins, they also inhibit lytic infection. So BZLF1 and BRLF1, the promoter protein, and EBNA1 is actually the only viral protein active in the latent EBV infection. So here we have, there's three different forms. Here we've got two shown in the slide. There's latency 3, latency 2, and latency 1. Latency 3 would be the most active amount of proliferation, with latency 1 essentially being dormant in the host. So here we have latency 2. There's an activated B cell, and they're moving into the memory B cells. And this is how the virus survives, which I'll get to in a little bit. What they do is they evade the host immune system by basically avoiding detection. In plain terms, they're hiding in the host. So here we have in latency 1, there's virtually no gene expression. And in latency 1, the virus can lay dormant in the host for years and years. And there could be, over time, this could eventually lead to oncogenesis, which I'll get to a little bit at the end. So here we have the EBV capsids. As I mentioned before, here's the tegument, which is made up of some late proteins. Here we have the genome, the envelope with the spikes, and we have the portal vertex, which is kind of a unique trait of the herpes viruses. This is where the genome enters and exits out of the capsid. So the genome will come out or go in. And from research, I saw that it's actually very important too in just maintaining the DNA in the replication processes. So here, move my face. We've got the capsid, or the envelope as a whole, the whole entire virion. And here you can see it's an icosahedral capsid with the genome inside. And then we've got the proteins, packaged RNA. So another thing to touch on is that EBV, while it lays dormant in the host, it can have a lot of oncogenic potential. So Hodgkin's lymphoma and Burkitt's lymphoma are two of the cancers caused by Epstein-Barr virus. Half of Hodgkin's lymphoma cases are said to be caused by EBV. 20% of Burkitt's lymphoma cases are caused by EBV, and almost all of endemic Burkitt's lymphoma cases are caused by EBV. Endemic Burkitt's lymphoma is a cancer seen in equatorial Africa. From what I saw, it's mostly affecting children, which actually makes it the most common childhood cancer. So what EBV does to survive in the host is it's going to remain at low levels, as I sort of touched on already, and it's going to target memory B cells. And what the virus is going to do is it's going to prevent the antigen presenting cells from presenting the virus to T cells. And it's going to use the viral protein EBNA1 for that. So EBNA1, it's going to prevent MHC1 antigen presentation. And the mechanism that it achieves this by is just, it prevents unfolding of the 19S proteasome. And this causes no replication, and the viruses will remain inside the memory B cells at this time. EBV, another way that it avoids the immune system is it prevents the signal transduction. So what it does how it achieves this is by LMP1, and what LMP1 does is it prevents the phosphorylation of STAT1 and STAT2, and this also blocks the antiviral effects of IFN1, which we saw in class. So this causes T cells not being proliferated, and it allows the virus to remain latent for extended periods of time. So here I just kind of touched on how this virus can cause cancer. So the oncogenesis. EBNA1, it can promote LMP1. So these are also ways in which the virus avoids the innate immune system, but all of these proteins seem to have different functions in this virus, which made it interesting and challenging at the same time. So EBNA1 promotes LMP1, and this results in cell proliferation. And nuclear proteins of EBV, they activate cyclin D2 and CDK4, which basically what this is doing is it's just pushing the cells through the cell cycle from G1 to S phase. Another protein, EBAN3C, can directly bind to P53, which we know is a tumor suppressing gene, and this prevents apoptosis.